Hello and welcome to today's lecture, which is on nociceptors and the axon reflex. So by the end of this lecture you should be comfortable with the nociceptor and the types of stimuli it can detect. Nociceptors are also known as noxious stimulus detectors. How they differ from innocuous fibres in terms of stimuli, receptors and transmission speeds. How thresholds of firing are not fixed, and we'll compare this with the all or nothing principle of cell physiology. The axon reflex and its role in inflammation, and finally the similarities and differences between acute inflammation and neurogenic inflammation. Okay, so just to recap on the different classifications of sensory nerves, we talk about A beta fibres, which are these large diameter, large myelinated innocuous fibres, so they detect light touch such as stroking, punctate stimulus, etc. And these have very fast conduction velocities due to the large diameter and the thicker myelination, and can conduct in the range of 10 to 100 meters per second. We talk about A delta fibres, which are much smaller in diameter but still myelinated, albeit with a smaller, thinner layer of myelination. And again, these are involved in the sensation of touch, more to do with the stimulation of hair follicles. But they also have a role to play in nociception, in mechanical pain as well as heat and cold pain. And they also have a non-noxious role in detecting cooling. And then we talk about C-fibre. The picture you see here is actually of several C-fibres housed in a Schwann cell. So we talk about Schwann cells not only being involved in myelation, but also being non-myelating and housing C-fibres. So an unmyelated fibre will also be covered in a Schwann cell. And this little bundle here is called a Remak bundle, which is several C-fibres within a Schwann cell. So we talk about touch being an innocuous stimulus, which could be mechanical touch or heat or cool. And then we talk about nociception. So we see that C-fibres are predominantly nociceptive, although they do have a role to play in the detection of pleasant touch and as well as warm sensation. If we look at the conduction velocities, here we see a setup whereby a stimulus is applied to the skin and then nerves are recorded a small distance away. And we see that with initial noxious stimulus, we get a huge firing of the A alpha and A beta nerves, just due to the purely mechanical deformation of the skin, so not noxious, just the movement. Then straight away we get an A delta firing. This is not as high, not as big an amplitude, but very close to the initial A beta firing, which tells us it's got a very fast conductance. And then there's quite some distance before the C-fibre fires, and that's or at least reaches the detector, and it's of a much lower amplitude. So C-fibres are much slower and much smaller amplitude, but that has a specific role to play, as we shall see later. So if we look in the dorsal root ganglion, we'll see that it's made up of several cells. We can split these very basically into large cell bodies, which belong to the larger A alpha and A beta neurons, and the smaller cell bodies, which belong to these A delta or C fibres. We can then stain these for several different ways. One of them is staining for non-peptidergic neurons, which are the green ones here, and then peptidergic neurons, which are the blue ones. As we shall see, peptidergic neurons are very important when it comes to inflammation because they are able to release neuropeptides from their peripheral afferents, their terminals, and these are things such as substance B and calcitonin gene reactive protein, which is CGRP, and that is very much involved in inflammation. So if we divide the proportion of the nociceptors in the dorsal root ganglion, we'll see that about 40% of them have myelination. That's A beta, A alpha, and A delta. About 40% are peptidergic, which release the substance B, as I said, and the CGRP. And another 30% are non-peptidergic. So this tells us that A beta fibres are roughly 20% of the dorsal root ganglion cells. A delta, which are both peptidergic and non-peptidergic, but are myelinated, take up 20% of those nociceptors. And then the C-fibres are about 60% of the dorsal root ganglion cells. So you see that nociceptors, ignoring sort of 5% that code for pleasant touch and warm, but 55% of the cells and the neurons in the dorsal root ganglia are just for pain. And if we add the A delta ones, we get a number closer to sort of 70, 75. So nociception is obviously a very important mechanism, okay, and it's integral to survival of us as an organism. So if we take a look at low threshold afferents for a minute, these are the A beta ones we just talked about, we'll see that they have end organ receptors. Okay, so their A beta nerves will go into a complex end organ receptor, which is made up of separate cells or numerous cells, depending on its construct. These are, for example, the mycinous corpuscle and the Pacinian corpuscle, which detect sudden change in movement, and the Ruffini's corpuscles and the Merkel's discs, which detect slowly adapting movement, so slow change and increases in pressure within the skin. So these are all complex end organ receptors made of separate cells from separate embryological tissue that envelop the A beta nerve. Okay, so when we detect mechanical stimuli, it's going through these receptors and then into the A beta nerve. This is very different to nociceptors, which are free or bare nerve endings. Okay, so they have no specialised end organ receptors, but they do, however, have molecular receptors in their cell membrane. Okay, so when we talk about receptors in nociceptors, we're talking about molecular, on a very small molecular level, rather than these large macroscopic end organ receptors. So this picture here just shows how nociceptors are free nerve endings, and we see the red arrows are pointing to the very fine tendrils of a C nociceptor, an unmyelinated nociceptor. And you can see how it goes all the way to the outer part of the epidermis. Now remember that the blood supply to the skin is purely in the dermis, and the epidermis doesn't have any. So we can't have organs that are too big in the epidermis. So these C-fibres go all the way to the end. Now obviously this is very important, because if we're going to damage tissue, we want to detect it very, very quickly. So it seems to be that the nociceptors are very strongly linked not only to tissue damage, but to the keratinocytes. And the keratinocytes, skin cells, seem to play quite an integral role in coding for noxious stimuli. But that's quite a modern concept. So just for now, remember that the nociceptors are free nerve endings, and they go all the way to the very superficial parts of the epidermis, whereas the A beta fibres are more to do with cell organelles and lie slightly deeper. Right, all sensory nerves have a receptive field, and when we talk about receptive fields quite a lot when we're talking about hyperalgesia and allodynia and pain hypersensitivities. So the receptive field of one neuron is very small, between 2 and 10 millimeters in diameter, and this diagram here shows several sensory nerves, and the one highlighted in red is just showing you that receptive field of that one. So you can see how the adjoining nerves overlap. So if you had a loss of that nerve, giving you a sensory loss, it'd be very hard to detect due to the overlap. But if you have an increase in sensitivity of that nerve, then that whole receptive field will become hypersensitive. Now that's the receptive field of the peripheral afferent fibres. Okay, but these peripheral afferent fibres all converge on a second order neuron. So in the dorsal column system, that would be in the medulla, but in the spinothalamic tract, that would be in the dorsal horn. And you can see how numerous receptive fields of all the different peripheral afferent fibres will converge onto one second order neuron, which gives us the larger sort of light red circle, which is the receptive field of the second order neuron. Okay, so we go from the receptive field of a neuron, which is very small, and that summates into a larger receptive field for second order neurons. And in fact, by the time we get to the thalamus and up into the somatosensory cortex, we have whole body receptive fields on individual neurons. Okay, so you could argue that each cell in the thalamus has a whole body receptive field. Okay, that's in part true, but by the time we get to the somatosensory cortex, it's very much true, and that's how we get the very quick change in the size of the homunculus, or the size of the discriminative ability of different parts of the body that alter throughout the day. So sometimes we talk about braille readers who, when they go to work and they read braille 9 to 5 as part of their job, half an hour before getting to work, their somatosensory cortex actually alters and changes to make their fingers more discriminative, more able to pick up the reading. And then when they finish work and go home, that's the homunculus actually readapts. So this very fast plasticity of the brain is due to the specific recruitment of different receptor field neurons. Okay, so it gets rather complicated, but just remember that the peripheral afferents have a fixed receptive field, which is unchangeable. But when you go into second order neurons and third order neurons, the receptor field is very much changeable, and this is via a complex mode of inhibitory and excitatory interneurons. They will cut off certain stimuli to those nerves. Okay, so just showing a bit more detail here, this diagram shows the receptor fields of individual neurons, and on top we see the leg, and then it goes down to the foot, and the very bottom the toes. And you can see quite clearly how the receptor field is much larger the more proximal we are, and as we go more distally, the receptor field gets very small. So this is an important aspect that allows us to have better discrimination. Obviously the areas of tactile sensitivity, such as the fingers, have smaller receptor field, and therefore you fire more nerves off depending on the size of the object, and that will help some way towards discrimination. The other way we discriminate more clearly is via, again, a very clever mode of lateral inhibition, which shuts off surrounding neurons and focuses or highlights the central neurons that we are focusing on. Okay, but you can see the receptor field, how it gets smaller as we go distally, and if we want to see that clinically, we just merely have to look at something such as herpes zoster or shingles, and you'll see that each of these blisters is actually the receptor field of an infected neuron. Okay, and these would be both A beta fibers, but predominantly C fibers, thus giving us that sort of shingles pain, the hepatic neuralgia. Right, now, nociceptors only respond to 3 types of stimuli, okay, in very broad categories. And if we look at this illustration from the Explain Pain book by the Noy group, we'll see how we have mechanical stimuli, which will activate mechanical receptors, molecular receptors on the nociceptor, and that will set off an action potential, or at least it will release ions into the cell. We have thermal receptors, which detect heat or cold. And then we have acid sensing channels, which can pick up acids, protons, etc., as well as a vast array of chemicals, which we'll look at later. But these receptors are very specific to their stimuli. So a thermal stimuli will not be activated by an acid, and similarly an acid channel will not be set off by a mechanical stimuli. If we look at the different types of nociceptors, so we've already mentioned we have A delta, which are the unmyelinated, but fairly small ones, and then we have the myelinated, fast conducting, and we have the C fibers, which are unmyelinated and therefore much slower. We'll see that the A fibers evoke a pain that's often described as prickling, sharp, or aching. This is quite quick pain due to the fast conductance. And these are divided into 2 types. Type 1 are polymodal and respond to all 3 stimuli, so heat, mechanical, and chemical. Whereas type 2 have a very high threshold to mechanical stimuli and are known as mechanically insensitive afferents, or MIAs, or silent nociceptors. So these have an extremely high mechanical threshold, much higher than normal. So we know that nociceptors already have a high threshold, and they're known as high threshold afferents. These have an even higher threshold. So to all intents and purposes, they're mechanically insensitive until they become inflamed and sensitized, and then they become very much involved in detecting mechanical stimuli. And just to remind you there that A fibers are also detect cooling sensation, so non-noxious cooling sensation. Okay, and then the types of C fiber, the majority of C fibers are polymodal. We call them CMH, which is C mechanical heat, and they evoke a sensation of burning pain or itch. We also get these mechanically insensitive afferents, known as silent nociceptors, and similarly with the A fibers, they respond vigorously to chemicals such as histamine and play an important role in inflammation. And again, just to remind you, we have these low threshold C fibers that exist that detect pleasant touch as well as warm sensations, so non-noxious heat. Right, a little experiment here. If I could ask you just to take a bit of sensitive skin from your inner arm and just pinch it gently between your thumb and forefingers. Okay, and then just give it a really hard pinch, as hard as you can bear. Okay, I'm not sure what the health and safety is over there, but as I'm in England, I'm not too concerned. So pinch really hard enough to cause pain, basically, and then sit back and wait. Now, if you've done this hard enough, you should have felt a really intense initial burst of pain, and then a slow, sort of much milder but slower build-up of sort of dull aching afterwards. Again, if you're not doing it, if you're not feeling that, you're probably not doing it hard enough. Okay, so what we're seeing here, and we call this first and second pain, we're seeing initial spike, which is caused by the A delta fibers, and then a slower build-up of second pain, which is activated by or caused by the C fibers. We can tell this experimentally, not just by the speed of conduction, but if we block the A delta fibers, and you can actually do this with a pressure cuff around the arm, which will take out the larger fibers preferentially, and then activate it, you will see that we lose that first spike, but quite importantly, the bigger second pain, the slower second pain is bigger than it was initially. And if we take out the C fibre, which is the bottom diagram, we'll see that we get a first sharp spike similar to the first experiment, and then nothing, which tells us clearly that the second pain is C fibres. So we know that the first pain is A fibre, second pain is C fibre, but that the first pain actually inhibits or reduces the second pain. So A delta goes some way to inhibiting or reducing the degree of second pain. Okay, the slow kind of diffuse build-up of pain. Right, and this is partly because faster relaying L data causes first sensation, first pain, and that's because they recover quicker. So you can get isolated, discrete pains, and that's going to localize quite quickly to your cortex, whereas a much slower delayed sensation comes from the C fibers. And interestingly, if we look at the functional MRI scans of the brain, we'll see that along the top row, where it says S1, this is the somatosensory cortex, we'll see that it's highlighted mostly when the A delta fires. The second pain of that graph is not really happening there. You see a sharp spike and then it sort of disappears. Whereas the bottom two pictures are S2, which is the supplementary cortex, and the ACC, which is the anterior cingulate cortex. And these are very much about emotional processing of pain. And we can see that the second pain in those graphs is much higher, okay, almost as high, and in the bottom one's higher than the first pain. So we're getting this picture whereby the initial fast localization of pain goes to the somatosensory cortex, in part via the fast-acting A delta fibers, and then the supplementary cortex, which is kind of mixing the sensation and the discrimination with the other parts of the brain, sort of responds to both the fast and slow pains, whereas the cingulate cortex, which is very much about motivation and the affective aspect of pain, almost has no activation via the fast A delta, but very much so with the slower C fibers. So we're getting a picture whereby the slower build-up of the C fibers is much more about the emotional aspect and a sort of homeostatic role of pain perception rather than just local discrimination. And these are the pathways just to explain that. So we have the lateral pathway, which goes via the first and second order neurons synapsing in the lateral thalamus and then going to the somatosensory cortex. This is that fast discriminative route. And then we have that medial pathway via the medial thalamus, which then goes into various parts of the limbic system, which is that slow affective aspect of pain. And when we say the limbic system, we're talking about the hippocampus, the cingulate cortex, the amygdala. And these will be going through these areas and sort of running past any memories. Have we had this pain before? What did it mean to us? Was any fear or anxiety involved? And it very much packages up to determine what our response should be. So the somatosensory cortex tells us that the pain was on our arm, whereas the limbic system will tell us that this pain is very minimal because we're pinching ourselves and we're not too bothered, or that it could be something horrendous such as a snake because we've been bitten previously. Right, another important aspect of nociceptors is that they can be inhibited by low threshold mechanoreceptors. So the low threshold mechanoreceptors are the A beta fibers that we're talking about, nociceptors being the high threshold fibres. Okay, and in this diagram we see how a nociceptor is it's able to stimulate a projection neuron via an excitatory interneuron. Okay, so here we have nociception going via an interneuron, contacting or stimulating a projection neuron, which will then fire and go up to the brain. We also see how these low threshold mechanoreceptors have direct synapses with these projector neurons. Okay, but that synapse is actually inhibited via an interneuron, a GABAergic interneuron, which will inhibit not just the low threshold mechanoreceptor, so cancelling out that interaction, but will also reduce the stimulus imposed by the nociceptor. Okay, and this is sort of the beginnings of the pain gate theory, where we see that a low threshold stimulation, such as light touch or stroking, can override or at least reduce the nociceptive stimulation. Okay, so when a child bangs their knee and their mum rubs it better, this is to some part what's going on. Obviously, it's far more complicated than that. But this was the beginnings of the pain gate theory, and was, in its time, a huge advancement in pain science. Right, so going back to the peripheral afferent, we have we've mentioned mechanical and heat as being stimuli for nociceptors, but inflammation and inflammatory mediators are there's a huge array of these. So nociceptors correspond to so many chemicals. We have histamine and prostaglandins and serotonin being released from mast cells. We get ATP, hydrogen ions, acid, glutamate, adenosine, all these things released from tissue damage and platelets. And then various immune cells can release things like bradykinin, nerve growth factor, interleukins, so all these cytokines and neuropeptides, which can also activate and set off nociceptors. Okay, but it's important to remember that all these receptors are molecular receptors. Okay, don't get them confused with the larger cell organelles on the low threshold mechanoreceptors and the A beta fibers. Now, if we zoom into this, we can see that along the distal sort of surface of the nociceptor, we have all these channels for these receptors. But notice how they're not ion channels. Okay, these are not ion channels that allow the influx of sodium, but are actually a different type called gene-protein coupled receptors. And what they do is, when activated, so if we look at prostaglandin here, that activates a specific prostaglandin receptor, the EP receptor, and that will actually release a kinin, a sort of chunk of protein, which will go via a complex cascade of enzymes to eventually alter the protein makeup of the ion channels. So we see a method in which the mediators of inflammation set off an enzyme cascade which alters the makeup of the ion channels, and that sensitizes them or reduces their threshold so that a much lower concentration of acid or a lower thermal stimulation is more likely to set off. It will allow greater influx of ions into that cell and make it more likely to set off an all or nothing action potential. Okay, so that's the method by which we get sort of sensitization of peripheral afferents, is via these chemical mediators. Now, another thing we learn at school is that sensory nerves only fire in one way. Okay, if you remember, this is mainly in part to this put down to this sort of all or nothing action potential, and when the threshold is reached and it fires, when you reach the peak, it's sort of plus 30 millivolts, and then reduces. We see this sort of hyperpolarization or the repolarization, and then the hyperpolarization, and this is called the refractory period. And that refractory period theoretically cannot be overridden, and therefore an action potential can only propagate in one way. If you stimulate it from either end, it can be activated and propagate in both ways, but the way the synapses work and the nociceptors are that they can only get stimulated at one end, and therefore the axon is only going to go down one way. Now, unfortunately, this is not true, and the nociceptor is extremely clever in this department. What we see if we get activation of nociceptors by tissue insult, so in this case we're damaging the skin, we're getting bradykinin, serotonin, prostaglandins, ATP, all of this released from the damaged tissue cells and keratinocytes, as well as mast cells degranulating and releasing histamine to that nociceptive terminal. And we see the normal activation of an action potential, which goes down towards the cell body in the dorsal root ganglion and onto the second order neuron. Now what we also see is we get an antidromic impulse that goes in the opposite direction. So an antidromic just means the opposite to orthodromic, so they're going down the wrong way or backwards, and it goes back down towards the terminals, leading to release of neuropeptides at the periphery. So hopefully at this point you're remembering the different types of C-fibres. We talked about the peptidergic versus non-peptidergic. So this is that percentage of C-fibres and A-delta fibres that have neuropeptides as a neurotransmitter. So these can have this antidromic impulse, which we call the axon reflex, and that will release these neuropeptides into the periphery. Okay, this leads to what we call neurogenic inflammation, so inflammation caused purely by the nerve cells. What we find is that these neuropeptides, so substance P, CGRP, are housed in vesicles at the nerve endings, and when the nociceptor is activated, they get released into the tissue in the terminal and into the terminal axons of the other branches of that nociceptor, and these will act on capillaries, mast cells, and smooth muscles. So we get a vasodilatation as the arterioles dilate. This will give us a sort of a red flare response that we see in the skin. Then we get the plasma extravasation, or a wheal, around the burn or the tissue injury, and that's as fluid leaks out of the postcapillary venule. You'll also see smooth muscle contraction, okay, preparing to heal the injured or potentially injured tissue, and we get mast cell degranulation. So here we're having release of neuropeptides from the nociceptor terminal, activating mast cells. The mast cells then degranulate, releasing histamine. The histamine, which we know is algogenic or is capable of stimulating nociceptors, will stimulate the nociceptor, causing it to release more neuropeptide, substance P, etc., which will cause more mast cell degranulation. So we see this cycle whereby tissue damage leads to activation of nociceptors, which leads to more activation of mast cells, leading to more nociception. So nociceptors play an integral part in a sort of feed-forward or positive feedback loop to enhance and exaggerate the tissue and inflammatory response. Okay, now this is essential to get the amount of arterial dilatation, get the neutrophils in to fight any microbes, to get the healing process underway. And as long as the tissue heals normally, that circuit gets broken, less noxious stimulation, less neuropeptide being released, less mast cell degranulated. At the same time, the tissue's healing, so you get less keratinocytes and mast cells releasing things there as well. And as it all winds down slowly, the nociceptor becomes quiet and stops being activated. Okay, so we see that during acute inflammation, where we get erythema, edema, heat, and pain, we can get all of these signs and symptoms from release of nociceptors, activation of nociceptors. Okay, so generally they're completely linked with acute inflammation, and you can't separate the two. But in some instances, we see activation of nociceptors in the absence of tissue damage, which can lead to a mild acute inflammatory reaction. Not to the extent of tissue damage, because it won't be as bold and brash as a full-on inflammation, but to a very subtle level, we still get the same sort of stuff. Slight change in tissue texture, trophic changes to the tissue, and this is becoming very well studied in the regions, in the areas of things like eczema, where skin changes occur, interstitial cystitis, whereby the change in the bladder mucosa is seen on biopsy, but there's no bacteria or any causative agent, and they believe there's a lot of backfiring of these nerves, releasing neuropeptides into the mucosa, causing this. So we'll come back to this during the weekend on pain, because it's a very important principle. Right, just want to show you a video just to help you cement this in your memory. So activation of a nociceptor will fire the normal way, the orthodromic way along the axon towards the central nervous system. But at the same time we get this axon reflex whereby action potential is propagated backwards down the branches of the nociceptor, and this results in the firing of neuropeptides from the terminal endings, which will activate mast cells to degranulate, open up blood vessels, releasing plasma from these blood vessels, as well as activating smooth muscle. Okay, at the same time we can get a backward action potential all the way from the central nervous system. This is often described as a dorsal root reflex. Okay, it's very hard to determine experimentally, but they have done it, and there's lots of schools of thought that this is occurring a lot of the time. Anytime that you get a referred pain phenomena, you'll probably get a referred activation of nociceptors and therefore a chucking out of neuropeptides into that tissue. Now this is more anecdotal than hard science, I'm afraid, but if you look at someone with a hip pain, for example, that's getting referred pain into their knee, eventually it becomes a little bit boggy, a little bit warm, a little bit sensitive, and we may think there's something wrong with the sort of lateral collateral tissues there, ligaments, etc. When in fact probably you're getting an antidromic dumping of neuropeptides from that referred pain. I think it's one of the reasons why doctors get confused quite often by referred pain, because of the local tissue changes. Right, so hopefully from this lecture you're now comfortable with the nociceptor and the different types of stimuli it can detect, how they differ from noxious fibres in terms of stimuli, receptors, transmission speed, thresholds of firing, the axon reflex, and then neurogenic information. Okay, so thanks very much, and I'll see you for the next lecture.